Thanks for your introduction. Hello, everyone. So my name is Mamunur Rashid. I'm working as a scientist at Bioinformatics Institute under STAR. So my presentation title is a multi-class supervised sparse canonical correlation analysis on multimodal data for predicting breast cancer treatment response. So let's see where actually the prediction is important. So currently ongoing, right, when one patient actually diagnosed as a cancer patient, so based on the expert knowledge, so all the patient actually are assigned as a common treatment. So on this fact, the result is a few patients response well, is effective, as many patients non-effective, and even some patients is a very worst case. So the future medicine working for to tailor the treatment for individual base, so that before go for treatment, we can decide which patient can be improved or effective using the traditional, the existing drug or not, or which patient can be go for the further combination therapy. So based on future medicines will try to use the different data sources in terms of multiomics data. So in the future medicine vision, so different data sources was as an integrated manner to subtype the patient is a specific cluster. So each patient, its cluster can be treated as a specific treatment so that this can be effective for all the patients. So when there come data, so nowadays, based on the advanced technology, multiomics data become more available. So why multiomics? So if you think the whole system is a human body, so each part is a certain omics data, certain omics layer. So when you work in genomics, transcriptomics, proteomics, that means we are still playing in a certain level, not the whole system. So if we connect in a way in a network, all the layer, then we can get the global view, and it will help to understand the complex phenotype like in breast cancer. So how to handle this large amount of data, the different omics layer, because they are very different in terms of number of statistical properties, number of phenotypes, and the different types of character of the omics nature. So we do one review in the literature review. So based on the development method, there is several methods. Most methods do the similar thing, but their methodological is different. So based on the methodology, they can be largely categorized as early integration, late integration, and intermediate integration. So early integration is very simple, like you have a different data source, you can connect them and treat it as a single omics data. But difficulty is the large number of dimensions. So it's very hard to analyze the data in this way. But late integration is getting focused in the deep learning case. But in the deep learning case, in terms of late integration, you apply your method individually is omics level, and then you integrate in a result section. So it's very hard to interpretable, but this work very well for classification manner. But in terms of intermediate integrations, there is a two specific feature, like this method actually basically based on the data dimension reduction. So you can visualize your findings, and also the factors you obtain from the intermediate integration, like SCCA, multi-omics factor analysis, like below. So this factor can be used for your downstream analysis. So I found that most of these methods develop based on the clustering of the sample, like cancer type, but they are hardly tested for response prediction in terms of treatment response. So I try to summarize the current challenges, like accurate prediction is always challenging. Second, lack of robust multiomics integration algorithm, because existing method, they can categorize the breast cancer sample is a 5 subtype. But when you consider is a phenotypic response in terms of a treatment, so one treatment response can consider include all of the subtype. So it's very hard and challenging how to tackle different response subtype. And currently the existing multiomics method actually, they use a different omics level, but it's hardly they include the cancer extrinsic feature. Cancer extrinsic in terms of like tumor, immunomicroenvironment, and path activity levels. And they also exclude the interaction, because in terms of multiomics, we're thinking multiomics features are always connected in the network. So they belong in the interaction. So, and interpretability is very challenging in terms of multiomics feature. So our objective is to develop an interpretable multiomics machine learning method to predict treatment response for patients given their pre-therapy omics data, and to identify novel multiomics biomarker for its response test. So context, so we can use this for a prognostic or diagnostic cases. So recently this paper has published actually. So we interested this work based on this paper. And this paper, they reported as a large number of clinical data. So we consider this one. And I summarize the data in here. Like there is a different level of information like clinical. In terms of DNA, there is a mutation status, also RNA expression. From RNA expression, we use the whole transcript for first identify the tumor immunomicroenvironment features, as well as the pathway activity score. So the raw dimension of the data is like 147 was the common samples. Individual means here's the patient. And different number of dimensions. So we do some data quality control. So we finally get a reduced data dimension. So for quality control, we restrict some criteria like the mutated gene should include at least 10 samples. So it's reduced, like 35 genes can be used for analysis. Same as for, we do like a differential expression for, because the transcriptome is a very high dimensionality. So we mostly include the most informative one. Also, so finally, the reduced data we use for the phenotypic response prediction. Like we have 4 different category of phenotypes. Like in terms of drug response, we have a PCR and RCB. PCR means you apply the treatment and the patient actually good response. So that's called the pathologically complete response. And RCB also categorized in the 3 category. So the RCB one can be considered they are good, not best. And RCB2 is a moderate, and RCB3 is almost resistant, so it's not effective. Okay. So this is our method. What we try to understand. Like we say, like the multi-class. That means we consider the response classes that is indicated in our objective function. So our objective function has a 4 part you can consider for understanding. Like C means we consider the class level in our objective function, and M is a magenta color, is consider the different omics level and different data. So we use a dimension detection table that's called the intermediate integration. It's called the CCA, canonical correlation analysis. So it's actually ensure the interaction between different data modality. And we have the another part is called the sparse constant. So it's still the data is a high dimensional. So we try to use the sparsity constant so that the non-informative feature can be excluded from our estimated feature vector. So this is actually the summary, what we try to say. Like we have the 4 steps here. First step, we have the we need to data gathering from the multidimensional data, multimodal data. So we have the like breast cancer synapses, and we collect the different information like DNA, RNA, and also clinical information. So we have the response category, and the data, yeah, you can see the different color. Different color present the different response category actually. So in step 2, clinical data is often heterogeneous in terms of the number of samples. So in our main algorithm, we try to engage the colors balancing technique. So we use the one versus all technique so that our method can use the oversampling technique to solve the problem. Later we say the MS2 CCA. That is our proposed algorithm. So in step 3, it can detect the response specific feature. So in our case, PCR related, RCB related, RCB1, RCB2, and RCB3. Finally, we feed this extracted feature in the machine learning classifier, we call say SVM. Then we evaluate the performance of the model. Okay, let's see. So for comparison, training our model, we divided the data into 70% for training and 30% for test. Test means the data never seen used in the model is a separate. So for comparison, I consider here 3 measures like AUC under the ROC curve, also AUC PRC in the physiological curve, and ACC measure. So based on, and for comparison, I consider here total 8 method. So in the figure, like y-axis is the measure criteria, x-axis is a different category level like PCR and RCB. And we found that our model has a superior result in almost more than 98%, 95% result we can gain from our model. So especially we focus on the AUC PRC curve, that is more reliable. We found all the cases our proposed model was much better than existing one. That means the outperform our model. So second question, actually we use different modality of data. So which modality is much more influential and informative? So we do the combination analysis like in the left figure. Then we use a clinical as our reference model. So we add DNA and we see for response prediction, the performance was improved. But when you compare with the RNA data, we see there is a big jump. That means it looks clinical plus RNA can be a most informative layer for predicting your model. Then in the right side figure, so we try to individually, there is no combination, and we see, yeah, it's true that RNA level was the most informative. And because the comparing the RNA level and all data was the same influence. So we think that there is some information that is missing in the DNA level, but RNA can carry that one. So now the question, why our model is perform better? So we try to answer here. We call it the interpretability of the model. So you can see there is a heat map. There is a 4 heat map like based on the RNA, DNA, tumor microenvironment, and also pathways. So the unique thing, since our model can extract the response related by the feature, so in y-axis you can see the response category, PCR and RCB, and x-axis is the detected influential feature that use our model to predict. So for example, in DNA, you can see the PCR, the influential feature is different. Some is common, but some is very different, distinct for subtype. So, but second question is the same as we can see the pathways. So the PCR has some in between common pathway, but almost many distinct pathway. So that means we can get here like tumor microenvironment and pathways and DNA and RNA level information for a specific subtype has a multi-omics interaction. That might be influencing our method to predict. So second question, can we actually understand this biology in terms of pathway level? How they different each other? Because we want to promote the precision biology. So I try to visualize the top 10 pathways detected in my method for a specific subtype. Then interestingly, I find they have some distinct pathways that is very unique, but they have also common. Like in PCR, the first pathway, antimicrobial peptide, also I found in the RCB2. So the PCR pathways, like antimicrobial pathway was positively associated, where I find in the RCB2, they are the negatively associated. Though they share the common pathway, but their regulatory role might be different. Okay, so is it true? So I try to understand, so I pick up a common pathway for PCR, then I try to visualize their multi-omics interaction network. So in this network, different shape of the nodes is represent different modality, data modality. Also different color is the different modality, like pathway, including the pathway, the genes, RNA, and tumor microenvironment features. So their connection interaction, actually the correlation that is generated from the data. And different color, so there is also indicated by different strength. And degree actually measure the number of connectivity for each molecule in the network. So I do the same thing for the RCB2 category, but interestingly, I can give you the clue here. Though these two categories share a common pathway, but the regulatory networks or module in terms of pathway might be different. That's I detected in my method because antimicrobial peptide pathway was positively upregulated in the PCR, but I find in the RCB that is a negatively upregulated. And you can see here, like this module is completely separated in the RCB2. But in terms of here, they have some influence and they're highly influential in terms of PCR. So that means their pathway is common, but their module of activations in terms of pathway is different. So if you want to individualize the patient, we can map in this network for each patient. That can be greatly improve the precision biology. Okay, so what I found in my method, so we found that in terms of classification measure like AUROC or PRC and SSC, our method, what we call like multi-class supervised sparse CCA outperform compared to existing method. And from the comparison of data subset performance, we found RNA with clinical profile was much more informative. So we use the multi-task or multi-class learning that enables the detection of context-specific, means the response-specific multi-omics interaction that include pathway and also tumor immunomicroenvironment information there. Also, our method can detect interpretable multi-omics feature with the biological pathways. Thanks.
Thank you, Dr. Rashid. We have time for some questions. Maybe I can start. So, sorry? No. Maybe I can start. So you utilize several different types of omics data to make the prediction, the drug response or treatment response. Would it be possible to try minimal set of such omics data to make the prediction, but still that's acceptable? Because it's quite challenging to get all those omics data from all the breast cancer patients. If you can make the same quality of predictions with a smaller set of input data, perhaps that's better in many ways. So can you comment on that?
You mean?
So if I'm correct, from the table you utilized some clinical data, and I can record the omics data, but you utilize several different types of omics data.
Yeah.
But would it be possible to use smaller set of such data
I think
in terms of the types?
I think you mean small means how small is that one?
No, I mean the smaller number of the omics data types. You can have transcriptome, genome, clinical data, all that sort of data, right? Mm. But you cannot get all those data from all the patients every time you want to make the prediction.
Yeah.
So I'm asking whether you can use the smaller sets of such omics data types.
I think it's possible because this is not a deep learning model, this is a statistical model. And the statistical model is quite flexible for a small data set. Okay? So based on your interest, it's possible, I think.
Yeah, okay. And, oh, yeah.
Hi. Oops. Hi, thank you for the great talk. Me and my colleagues were wondering if you could touch briefly upon the normalization techniques that you use, because in your variables, you have several different types of data, also because you derive them from so many different data types. So we also work with data integration, and we know that normalization is typically a critical point. So if you can give us some insights on how you did it in this particular case.
So in my case, actually, I need data normalization in terms of expression data. But DNA since 1.0, I didn't touch the data anymore. And there is clinical data, clinical data is almost unchanged. So mostly I handle the expression data, and the normalization is very simple. I took the log transformation, then standardization. That's all.
So there is, except from that, you don't really sort of scale the other variables that you use. You just take them at face value.
Yeah, so there's the facility in this method. It can handle different scaling data.
Okay, thanks.
Thank you.